Hello, everyone. Welcome to today's Odyssey. Have you ever wondered if lost functions or abilities could be restored after a brain injury? What if scientists could just print you a new brain piece to replace damaged areas? Well, researchers at the University of Oxford recently announced a breakthrough that brings this idea closer to reality. They have developed a way to 3D print neural tissues that closely mimic the structure and function of the cortex, the outer layer of the human brain. This is big news because the cortex controls our ability to think, speak, move, and sense the world around us. Damage here from things like trauma, stroke, or surgery can severely impact a person's quality of life. Right now, there are no effective treatments to repair or replace injured brain regions. But this new 3D printing technique using stem cells shows promise as a future personalized therapy. In this video, I'm going to explain what the cortex does, how these researchers printed cortex-like tissues, and what this technology means for the future of brain repair. There's a lot of cool science here, but I'll try to explain it simply without too much technical jargon. Let's dive in. Before we get into the details of the study, let's talk about how these researchers used 3D printing and stem cells to create brain-like tissues. 3D printing works by taking a material like plastic or metal and precisely layering thin cross-sections to build up a 3D object. But we can't 3D print with dead materials to make living brain tissue. We need special bioinks made of living cells and molecules. These bioinks need to be biocompatible, meaning they can interact with biological systems without harming them. They also must be biodegradable, so they can be gradually broken down and absorbed by the body after doing their job. One of the most promising sources of bioink material is stem cells. Now let's talk stem cells. These are master cells that can turn into any type of cell. Think of them like clay that can be shaped into anything. Some stem cells are present in embryos, others are found in certain tissues in adults like bone marrow. But scientists can also take mature cells, like skin cells, and reprogram them into stem cells. These are called induced pluripotent stem cells, or iPSCs. The technology to make iPSCs won Nobel Prizes in 2012. It lets researchers obtain stem cells from anyone just by taking a small sample of skin or blood and reprogram them into stem cells. The researchers used iPSCs because they can turn into any cell needed to build brain-like tissues. The iPSCs were first specialized into two main cortical cell types, such as neurons, cells that transmit information using electrical signals, and astrocytes, supporting cells that help neurons function properly. These cells were then combined into bioinks optimized for 3D printing neural tissues. The key ingredients our Oxford researchers used. All right, now for the fun part. How did the scientists actually 3D print these brain-like tissues? Imagine the researchers as chefs in a kitchen, and the stem cells are their ingredients. First, they took the stem cell dough and added some spices called growth factors. This told the cells to specialize into cortex cell types, neurons and astrocytes. Next, they mixed the neuron and astrocyte ingredients into 2 batches of stem cell batter, the bioinks. They added a gelatin, called hydrogel, to give the cells a sturdy scaffolding to grow on, kind of like the cake pan for our brain tissue cake. Then they loaded the bioinks into their high-tech oven, the 3D printer. It precisely layered and baked 2 cell-filled cortex layers representing different brain regions. Since our chef scientists had finished baking their stem cell dish in the 3D printer oven, it was time to taste test and see how close it came to mimicking real brain tissue. They first checked that their dish contained the right mix of ingredients. Indeed, it had very similar ratios of neurons and astrocytes to real cortical samples. Then they looked at how the ingredients were arranged. Under the microscope, the cell layers and positions matched what's seen in a natural cortex. Time to try a bite. They tested the electrical activity and found the signaling between cells mimicked native neuronal networks. To really sample the flavor, they tested if their dish could communicate with a mouse brain slice. Amazingly, their human stem cell creation integrated with the mouse tissue, forming connections and responding to stimulation. Overall, the tests showed the 3D printed tissue and a real mini brain had remarkably similar architecture, composition, and function. The stem cell dish was missing some ingredients and complexity, but still, what an encouraging first attempt. We could also use these 3D printed tissues as lab mini brains to test drugs without experimenting on people or animals. But don't get too excited just yet. These are still early days. The printed tissues were pretty simple and didn't fully replicate the brain's complexity. It's also super slow and expensive to 3D print human brain tissue right now. We're not going to see brain printing clinics popping up anytime soon, and we can't ignore the ethical concerns. Is it okay to engineer replica human brains in a lab? We need more conversations there. While promising, it's still very early, and many technical and ethical kinks need working out. But brain tissue printing has now gone from sci-fi to scientific reality. It'll be fascinating to see where it goes in the coming decades. And that brings us to the end of our odyssey into the world of 3D printed brain tissues. I hope you enjoyed learning about how researchers are using stem cells and 3D printing to recreate the complexity of the human brain. There are definitely challenges ahead, but also so much potential. It will be exciting to follow where this technology goes in years to come. Before we go, please subscribe to join me on more odysseys into groundbreaking research. Subscribing lets you bookmark this channel to catch future videos. Also, let me know your thoughts in the comments below. I want to hear your perspective on the possibilities and ethics surrounding engineering brain tissues. Until next time, stay curious.